Hello, everyone, and thank you for joining us. I apologize that this session couldn't go ahead earlier due to some technical difficulties on our end, but we are now joined by Andreas Jeromin, Chief Scientific Officer at COHEN Veterans Bioscience. He will be speaking on current status and future directions of blood-based CNS biomarkers. We will also have time for a short Q&A after the session, so if you do have any questions, please put them in the chat and we will ask them directly at the end. Thank you very much, and without any further delay, Andreas, I'll hand over to you.
Yeah, thank you, Chris, and good morning, good afternoon, good evening, wherever you may be as well. Pleasure talking to you. So I have 20 minutes, which what I call a little bit of a fly through to CNS biomarkers. Really significant progress has been made in this space as well, and I think we are, from my perspective, really at an inflection point. So I'm reporting today, or as the Chief Scientific Officer to COHEN Veterans Biosciences. For disclosures here, a little bit small, but I've been an advisor to Quanterix Corporation, who has developed the ultrasensitive single molecule array or similar technology as well, and I'll touch on this in one slide. Just to give you a bit of background on COHEN Veterans Biosciences, we are a nonprofit biomedical research organization, and the focus and the mission of the organization is to fast-track the development of diagnostic and tailored therapeutics for trauma-related disorders. So we recognize the value and importance of biomarkers, and it's one of the reasons for speaking here today. COHEN Veterans Biosciences has made significant investment into various platform technologies or programs. Again, for biomarkers, there is no magic wand, as I tend to call it. Really, what we do need is an integrated solution as well. And as you see on the slide here, there are various investment in the development of infrastructure ranging from a biomarker alliance, and I'll show you in the slide what that specifically means. That biomarker alliance is called RapidDX. Solutions for data sciences, not just for cloud-based data warehousing, but also for the development of analytics tools. That's done within the Brain Commons here, if you see my cursor moving. We have a digital health solution for wearables called Early Signal as well. Have developed elements for a turnkey clinical trial solutions, particularly adaptive in nature, and also developed a program to the lower left here around the development of really robust preclinical models, cellular models, which can be translated to humans as well. So on the next slide, I think from my perspective, this is one of the challenges we're facing as a community in the biomarker space as well. So we're not alone here, but others have performed these type of meta-analysis or landscaping analysis as well. And if you just focus to the right here, this is done specifically in the context of post-traumatic stress disorders, where we looked at various candidate biomarkers, including cortisol, BDNF, FKBP5, and others as well, like inflammatory markers, IL-6. And now you look at what the valency of these studies are. You see in green here either increased, in red decreased, or in blue no change. You can really find studies for these candidate biomarkers which really either report one of these three valencies here as well, either no change, or increase or decrease. And I think to me this poses a problem in this space, and there's really no, again, single cause for this, but there are multiple causes for these differences in findings. So in terms of gaps now, what really is applicable here and what has hindered somehow progress in the biomarker space is that the majority of these studies really are statistically underpowered, typically using small sample sizes for discovery as well, potentially also not in the population, which are really mostly of clinical aspects. So it's typically a case control design as well. I think one of the other challenges in the interpretation of the studies is really the level of validation of the analytical tool employed, so the famous assay here as well, and we'll get to this in another slide. Really having an independent understanding of its level of analytical performance, I think, is important, and that's what's captured here under reproducibility and best practices as well. And I think also fundamentally, we do realize that these are complex diseases and disorders. So what we do need is a approach which goes beyond the single modality, the single omics as well, to really start dissecting out the biological underpinnings of the disease, or what we call the taxonomy as well, in a framework of systems biology. We have launched, COHEN Veterans Biosciences and Alliance, a private-public partnership model as well, which we call RapidDX, which, as you see on the slide, stands for Research Alliance for Precision Therapeutics, Innovation and Diagnostics as well. That program now is, as I said, an alliance, a coalition to enable robust biomarker discovery, replication, and validation now as well. And as you see on the slide, there are various work streams now in place, ranging from biofluid assay evaluation to a biorepository we've established in the biosample hub. And then really at the core with biomarker work stream 3, which is focused about an integrated spoke hub model as well to enable an SAP, a statistical analysis plan driven biomarker discovery and replication model. And then 4, for the future, a de novo prospective biomarker validation study. And as you may have noticed in the chat box, there is my email address. If you're interested in additional details on RapidDX or anything pertaining to this publication, please feel free to get in contact with me. So again, to RapidDX, I think one of the key applications for biomarkers in CNS and beyond as well is really the development of this precision diagnostic toolbox and tailored treatments as well. And what is outlined on the slide here is really breaking down these clinically defined diagnoses into what we call biotypes. So using various machine learning, artificial intelligence approaches to identify clusters which are rooted in based on biology as well, and allow for the identification of biological mechanism, and then treatments repositioned or otherwise de novo discovered, which allow to specifically tailor the subpopulation based on a given biotype as well. And I think from my perspective, in CNS, but also other therapeutic areas, there certainly is a bit of a paradigm shift to now break down these diseases into subtypes or biotypes using various multimodal biomarker-based approaches. So on the next slide, just in again, high level here in this fly-through method, I should list or use as a title for the slide, the assay does matter. So what others and we in the space have executed and performed is what we call bake-offs here now as well, where we really do cross-platform comparison. This is now done particularly in the context of inflammatory biomarkers as well. And again, it's not so, the details now not so important. This has been published in Cytokinex, and I'm certainly happy to provide this publication. But the outcome of this is a report card, if you will, and this is modeled after the traffic lights, green, yellow, and red. So green is go, yellow is somewhere in between, prepared to stop, and red is not so good, basically. So if you just look at this high level, this has looked at various inflammatory biomarkers now in different platforms, one being Quanterix, Mesoscale Discovery, Myriad, this is the Luminex-based, bead-based assay, R&D Luminex as well, and then the Quantikine assay, kind of version of a high-sensitivity plate-based ELISA by R&D system. You see significant differences in performance, and various analytical parameters were assessed, and where green basically and a little bit of yellow is the top-performing platform. We have selected that platform now based on these analytical and other considerations to take forward within RapidDX for de novo biomarker discovery of candidate biomarkers, inflammatory biomarkers, and subsequent replication in other studies as well. So this slide here is an outline of what we call the Simoa technology. Again, the details are not so pertinent to the presentation today. But the point I want to communicate here is that the advances we've seen in CNS biomarkers, and this is particularly relevant to blood-based biomarkers now, has really been very tightly coupled to the development of these ultra-sensitive detection technologies. So the biomarkers which are related to the neuropathology as well, and I'll give you 2 case studies in the subsequent slides as well, are of really low abundance, even in cerebral spinal fluid, but even lower abundance in blood as well. And this really has required the development of these ultra-sensitive technologies. And I would say from my perspective, Simoa has really become the gold standard now for the detection of these low abundance biomarkers, typically in the lower picogram per milliliter and femtogram per milliliter range. The fact that this is also an automated technology obviously is important to enable really high precision assessments of these low abundance biomarkers. So an example I wanted to give here is on this slide, which is, you may have heard this earlier in the morning, neurofilament light chain. It's a member of a larger family of neurofilaments, which are different molecular weights, one being the heavy chain, the medium chain in the middle here as well, and then the light chain as well. And a number of publications, probably every week or so, you see a Samoa-based neurofilament light chain publication across the spectrum of CNS disorders and diseases. Has really helped us to understand the utility of neurofilament light chain in the context of a given context of use. As always with these assays, the antibodies deployed in these sandwich ELISA. So Simoa is basically at heart a bead-based sandwich ELISA platform with digital counting on the detection side as well. The antibodies deployed here to neurofilament light chain, this is in the so-called rod domain, have really been important for the development of these high sensitivity and specific ELISAs as well. Neurofilament light chain is a component of the axon, so it's considered a general marker of axonal injury or neurodegeneration. Here is a meta-analysis now. This is performed in CSF, but the data in blood look very, very similar as well. One of the excitements in the field of blood-based biomarkers really has been that there's an excellent correlation between CSF, NFL, and blood NFL, serum or EDTA plasma as well. Neurofilament light chain, as I said, is a generalized biomarker of axonal injury as well. So it is not really specific. In my view, it doesn't really have diagnostic utility as well, but it has become a treatment response or disease activity biomarker, probably headed or first demonstrated in multiple sclerosis, and there are other neurodegenerative and neuroinflammatory conditions where data point into similar directions. If you look at the slide here now as well, so you're looking from the left here, so these are corrected NFL levels in cerebral spinal fluid across a spectrum of CNS disease states. You have healthy controls to the very left here as well. And then you have to the very right here, very aggressively progressing diseases such as frontal temporal lobe dementia or ALS here as well, or Huntington's disease. This is here to the right, HD, and here a form of HIV-informed or induced neurodegeneration. And as you can see here, if you look at the corrected mean values here as well, the more aggressive and rapidly progressing the disease is, the higher the mean median concentration of neurofilament light chain are, both in CSF and in serum. Because this is a non-specific biomarker of neurodegeneration, neurofilament light chain has also been now coined or termed or labeled as the C-reactive protein for neurologists, or the troponin for brain injury or of the brain as well. So again, there's really a large body of evidence now across the disease state. I'll just give you two different examples here as well out of the Alzheimer's disease space. And this is, again, has made it into the public domain and various press releases and other forms as well. And what you see here as well is that neurofilament light chain in a form of what we call familial Alzheimer's disease. So these are patients which have a known mutation of Alzheimer's disease, which allows you to impute and calculate the time of disease onset. Neurofilament light chain in this particular cohort, it's the so-called DIAN cohort, was able to show increases 16 years prior to the calculated disease onset as well. And this is as early as some of the other bioimaging-based biomarker measures as well. This publication was published in Nature Medicine, I think about a year ago now, in the DIAN cohort. So the next slide, and again, I'm just trying to summarize here high level in the interest of time, is that we really have seen it, we as a field, this includes the spectrum of stakeholders in academia, industry, and biotech as well, have seen a transition from research use only applications for neurofilament light chain really into the clinical space, as well as a clinical so-called diagnostic marker. Again, as I said before, diagnostic, because of the lack of specificity, is not alone, at least, is not the utility of neurofilament light chain either in CSF and in blood, but it is faithfully reporting disease activity and treatment response as well. And as I said, this has been demonstrated in multiple sclerosis and now in several other disease states as well. So it is in clinical use as well to basically track responsiveness to disease and potentially tailor choices for these variously approved treatments in multiple sclerosis as well. And we will see very similar applications in ALS, already see this in ALS and other orphan and non-orphan diseases of the central and peripheral nervous system as well. Neurofilament light chain is not only expressed in the brain, so when I call this CNS biomarkers, I should really accurately call it CNS and PNS biomarkers, because it's also expressed in the periphery, and an active area of interest is really to understand the contribution of the PNS versus the CNS in those diseases such as multiple sclerosis and ALS, where both are affected. So again, in summary, thus far from the early days, and early days is less than 5 years ago, where the use of blood-based biomarkers now for various contexts of uses was considered science fiction. We really have moved from science fiction into science, into clinical applications as well. And I'll have a slide to what's still outstanding at the very end. So with this, I wanted to kind of switch gears a little bit from neurofilament light chain to a different class of biomarkers, which are centered around phosphorylated tau. So tau, again, is very closely linked in its phosphorylated forms to Alzheimer's pathology as well, as a majority of those already probably know. Alzheimer's disease has really seen advances from a purely diagnostically defined disease with neuropathological confirmation into a biomarker-based classification. And we call this classification the ATN classification. It's endorsed and developed by the National Institute of Health and other organizations as well. And ATN basically stands for amyloid as well. So this is a beta 42 and 40. T for tau. This is either what we call total tau or phosphorylated tau, and I will show you some data for phospho-tau 181. And then N, I would say the field or the community may be divided between either neuroinflammation or neurofilament. In my flavor, that stands for neurofilament again, but that's to be discussed and debated. So here now, again, just to summarize this, we have a biomarker-based taxonomy of the disease, based on pathological changes, which I would argue we can now reliably measure in the periphery, originally in cerebrospinal fluid, but now based on technological advances also in blood. So again, we've seen really a large number of publications over the last 2 years, and this is a very active area of interest, where various assays have been developed for different forms of phosphorylated tau. I will focus here on a specific phosphosite, which is known as phosphorylated tau 181. Again, I'm happy to provide you with some of the publications listed here. Again, this has been now one of these cases where we really have seen in the initial discovery and various independent validation and replication studies coming back to the importance of replication to really understand the performance of a biomarker. So again, just an example from some of these publications here. If you look on, this is tau PET imaging now as well, and here's a figure which has looked at the correlation of plasma tau 181 now to various degrees of tau PET binding. So this is a form of pathology, a measure of pathology based on molecular imaging as well. And if you look at the figure here, you see this is now further subclassified based on either amyloid status. If you look at all this phospho-tau in blood is highly significant to changes or to binding on tau pathology in the brain. And it's more so specifically correlated in the subgroup, which is amyloid positive compared to the one which is amyloid negative, if you see the cursor moving here. You can now group this in this more traditional area under the curve way, where you look at those which are either tau PET positive or negative. You see significant differences between the two. And based on the single biomarker alone, this is phospho-tau in blood now, you start seeing area under the curve which are clinically meaningful in the order of 0.9 or so. So again, just to pause here for a minute, we as a community can measure with these ultrasensitive technologies here now, phosphorylated tau, a form of phosphorylated tau in the blood, which allows us to predict tau pathology in the brain based on molecular imaging. This is tau PET imaging here as well. Also amyloid positivity based on amyloid PET imaging as well. It also, and this is not on the slide, predicts future cognitive decline. So here you have, with a single biomarker, the opportunity to predict and stratify patients, for example, for enrollment into clinical trials based on their amyloid or tau PET status. So where is this kind of all going? And this is a figure which Kai Bleno provided to me as well. And again, he provided this several years ago, and we all thought, this looks kind of something may happen 10, 15 years down the road, and it's actually happening now. It's happening in Europe now, it's happening in the US, and it's happening in Asia, it's happening global as well. So again, in addition to the clinical trial application I just described, one can envision here the use of these blood-based biomarkers for an initial screening approach. Are they above, are they normal, or are they below a certain cutoff? And I'll come to this in a second. And then require further clinical workup. So this can be done at a GP office, a generalized or general physician's office. It can be done in a memory clinic. It does not require a lumbar puncture, but based on this blood-based biomarker result, this would enable further workup, basically either by lumbar puncture or by other forms of imaging here. And you see lumbar puncture, molecular imaging, and volumetric-based assessment here based on MRI to the very right. So where is this all kind of going? And then hopefully you've been with me on this brief and abrupt, hopefully not too abrupt journey of progress and current status and future directions of blood-based biomarkers. Blood-based biomarkers are, as I said, and will continue to be implemented into clinical use and into clinical trials as well. I didn't touch on the regulatory component, but we're happy to address this in the Q&A in the chat box as well. Importantly, these blood-based biomarkers really reflect the underlying pathologies. Again, I didn't show you on these slides here as well, but there's various studies who have compared blood-based biomarkers to various stages of neuropathology, what we call BRAC stages in the field as well, both of neurodegenerative and neuroinflammatory disease states as well. There are actually ongoing trials who now use these biomarkers to make real-time stratification decisions of enrollment in a given clinical trial tailored around the mechanism of action. What is still outstanding to really broadly use these biomarkers is really a form of what we call assay harmonization as well. So the global biomarker standardization effort funded by the Alzheimer's Association is currently has undertaken a round robin and assay comparison I described for the inflammatory biomarkers for neurofilament light chain, currently embarking on a similar assay for phospho-tau. All of these immunoassays, they slightly employ different standards or what we call calibrators as well. So as we've done as a field for a beta 40T, the development of reference materials and reference methods to really assign an absolute value to this concentration is important as well. The other aspect to this, and I alluded to this in the previous slide, is really the definition of clinically meaningful reference intervals. And there are various studies underway, powered to 10,000 or so participants to define these reference intervals for various biomarkers. And it's also important to understand comorbidities. For NFL, as an example, we understand that age, gender, or sex determining NFL concentrations. So does body mass index to a certain degree, and potentially also extreme dysfunction of glomerular filtration or rate or GFR as well. All of this is basically underway, well underway, in order to enable really clinical use, a very broad clinical use of these biomarkers centered around a very given context of use. So I think with this, I think I'm good on time. Hopefully there are some questions in the chat box as well. Again, there's my email address, and there's also my email address on the slide. So with this, I want to thank you for your attention, and again, apologies for the confusion and the technical difficulties about the earlier scheduled presentation. Thank you.
Hello, Andreas. Thank you so much for that. I think that's all we've got time for. I wanted to thank you again for joining us. And if our audience have any further questions to you, they can send them to you directly through the conference platform. So thank you again for joining us, Andreas.
Thank you. Bye-bye.